All right, welcome to the second class, second of the Masterclass in Kaiser Neuromap. And this is the summer 2021, and today we're going to be talking about the history of neuroscience. The history of neuroscience, in some respect, can be kind of dry because it doesn't change that much. Occasionally we learn a little bit more, but it is useful to understand any phenomenon, any field of study, to know how it got to where it is. All right, so the first thing is, when it comes to understanding the brain and the mind, is where is it located? Where is our mind located? And most people today would say it's located in our brain, and that was an early observation that it seemed that the brain was the center of intelligence. But as often happens between generations, whatever the previous generation says, the next generation has more entropy. It's got to find a new view on it. It needs a flip if it's a dichotomous idea at some level. So we have Plato and Hippocrates saying that the brain is the center of intelligence in our mind. Aristotle, student of Plato, says, nope, it's not the brain. It's just a cooling mechanism. The human brain, he notices, is the most moist of all the animal brains out there, but it's just to cool the heart at some level, and heart is the center of intelligence and where our mind is located. And partly you can see that this might come about just from casual observation that when you get stressed, you can feel more activity in your chest than in your head. But we don't usually stick with that. Oh, I throw these things in just to show how little we know. That it may have been dinosaurs were roaming around, I don't know, 1,000 years ago, maybe even to this day. And we think, we have this, we're taught this grasp of our past as if it's all known. But the more we learn, we're probably living, at least in Civilization 2.0, that there was a civilization expanded much of the world, but then got destroyed by probably comets 12,000 years ago. And we built it back up, and we think we built it up for the first time, and it was actually the second time or maybe the third time. Just to let us know that our history is, in some respect, maybe not as fixed as we think it is until we know more facts. Okay, that was just thrown in there just to keep you thinking about what we don't know. So in some respect, I like to use the beginning of the history of neuroscience is trefonation. The interesting thing about trefonation, when you're drilling holes into skulls to relieve intracranial pressure, it's used to this day. So it started thousands of years ago, 20,000 years ago. We find skulls that have holes in them. Many of them have healing around the holes, which shows that they survived the surgery. This one probably didn't have survived. This one did. So this healing there, you can see the healing around it. And one of the reasons why trefonation came into existence, the first time what I always heard originally, it's the drilling holes to release evil spirits. Well, I don't think that's really what was the case. What it was is that out there when our ancestors were hunting mastodons and mammoths and other creatures, and they were chasing them, and they fell and they hit their head on the rocks. If they hit their head on the rocks and it was a closed head injury and the skull didn't fracture and things seep out, fluids seep out of the brain, the person often died. But if you hit your head on the rocks and you broke your skull and intracranial fluid was seeping out and so forth, you often lived. So the elders noticed, hey, okay, if you hit your head on the rock and you first you go unconscious and you don't wake up, let's go ahead and break your skull a little to relieve what's going on. And when they would drill in or open up the skull, often the patient would wake up, open their eyes. So very quickly got contingent reinforcement that this is probably a good thing to do. And they still to this day will put holes in skulls to deal with close head injury. The other approach is to put someone in an induced coma. Years ago, I was working on an induced coma device with a friend of mine who eventually needed that device. Unfortunately, we never got it built in time for him to need it, ironically. Okay, so the trepanation's been around. You can see it went to the Middle Ages, more the Enlightenment, more recent times. It's to this day. It's happening at hospitals around the world. Okay. Going back now, one of the most everyone knows about Plato. He is about Plato and Aristotle. Aristotle's like the first scientist or nationalist. He's the one who realizes to use your senses to figure out the world. Plato's like, don't use your senses because it's all sort of false. Try to think the abstract core aspects. And Plato's like our left hemisphere. Aristotle's very much like our right hemisphere. The right hemisphere deals with details, the configurations, the individual information, the events. The left hemisphere is the core. It collapses to the category. So in a way, if there wasn't a Plato and an Aristotle in the past, we would have to invent them to understand ourselves. So, but you have Plato in describing this triad of our soul, and then an empiricist doctor comes along, Galen, in the 2nd century. I think Northern Africa is where he was working there, and has another model again of a triad. And to this day, we still have, probably for good reason, that our mind includes our whole peripheral nervous system. So it isn't just the central nervous system in the head, and the heart does contribute. The gut does contribute. So it's at least it does resources, energy, and it's probably elements that are contributing to our mental process. And so we still have sort of a triad. So it likely is a triad at some level, but it's, again, the majority is our central nervous system in terms of how we respond to most things. Okay, so we go back to ancient times, and one of the theories they had is this theory of humors, that there are 4 types of humors, and depending if you have too much or too little, you'll exhibit certain characteristics. There's a picture of Hippocrates, the ironic thing. He looks a lot like Barry Sturman, my advisor and a business partner for many years. I was at a conference once when they put Barry's photo next to him. And Barry really did, create a foundation to neurotherapy that no one, if he hadn't existed, we would have had to make him up because it was so necessary to have that rigorous investigation of neurotherapy in the beginning. Okay, Hippocrates is a statement. Excuse me. From the brain alone arise our pleasures, joys, laughter, and jokes, as well as our sorrows, pains, griefs, and tears. Through it, in particular, we think, see, hear, and distinguish the ugly from the beautiful, the bad from the good, the pleasant from the unpleasant. Oh, my slideshow likes to move on its own. Okay, so Galen goes into the 2nd century. They have this belief that it's the fluids in the brain that's really the source of our sensory processing. So you have the front ventricle, deals with imagination and so sensory processing. The second one, ventricle, a chamber here, it says cognition, and the third one's memory. Sorry about that. All right. You can see trefonation going. I'm going to keep going on into the Middle Ages. The pictures, start getting a little bit more sophisticated as we hit the 16th century. Here's Leonardo, still kind of drawing the brain in a more primitive fashion. And then you get much more, you have dissection going on and getting much more accurate representation of the brain. Vesalius is the individual, again, empirically. It's all, you solve problems not through the Platonic thought, you solve it through Aristotle's approach, which is Aristotelian perception of the world and classification. It's all about empirical investigation trumps theory. Data trumps theory, as people would like to say. You get some data that doesn't fit your theory, you can't fix the data, you got to fix your theory. So Vesalius was attacked by his teacher. Again, the next generation always subverts the dogma of the previous one, and his teacher eventually became his bitter rival. Mostly because Vesalius just went ahead and did. He didn't just think, he did. He cut open corpses and saw what was going on there. Here comes I think, therefore I am, Descartes, who had a view of the mind and brain being a dichotomy. And my friend Joe Bogan, the late Joe Bogan, who was a neurosurgeon involved in split brain cases, said that Descartes could be viewed as the greatest scientist of the last 500 years because only a great scientist can make everyone think the wrong thing for so long. So the duality concept that Descartes created probably is a major fallacy, and then we got to realize there is no separation, though we perceive there is one often, but that's just a fallacy and a mirage. Okay, now we get into finally some neuroscience at some level, where we're getting closer to what we are, how we're going to address problems people have, mental health issues. With Gall, now a lot of people like to mock Gall even to this day because they don't really know much. Often we mock, but we don't understand. And Gall, his idea was that you could tell how much a ability was present in someone in the brain by how much that brain pushed against the skull and created dents and protrusions and so forth. The story goes that he had a friend growing up in school. This kid with really bulging eyes was very good at verbal ability, better than Gall was. And Gall took notice of it and goes, well, you know what? That kid must have more brain behind his eyes. That's why it's bulging out. And thus he realized, okay, then that must be where the verbal ability is stored in our head, is right behind our eyes. And that's the beginning of his idea. One of the great things he did, and which I do in my own research, is he did not try to figure out the brain mental function correspondence by looking at average people. That would be very hard. He went to the extremes, extreme skulls, extreme behaviors, criminals, insane. You find the extremes, that will help you find create a paradigm that lets you discern where the functions are that then can be applied to normal. But if you try to, look at the normal, it's like trying to look at different colors of white and eggshell. You can't see the difference, but you get the spectrum of all the functions in there, all the skull shapes. You can start seeing correspondences that you wouldn't necessarily see with those bland vanilla ones. So one of the models, they had 27 functions with different areas of the brain that were responsible. One of the problems is that the functions were too gross at some level, like sense of cunning, valor, fidelity. To be what we look today, we would break the functions down to something much more tangible, like grapheme phoneme conversion, how you look at a letter and convert it to a sound, and that would be handled in this area, or dealing with anxiety and the amygdalas and how they or fear, and we reduce the functions to these more manageable levels, and that's really about the only difference. Plus, we're looking at the brain now, not the skulls, because that has very little correspondence, the skull shapes, to what's really going on within. Now, another sort of positive step forward by Gall is that over on the left, that's how brains is. They started looking at the brains and saw all those convolutions. They said, okay, let's just draw convolutions. It looks like the small intestines. Let's just put the small intestines in the skull. And that's how a lot of pictures were for many, many centuries. When they started noticing the convolutions, Gall realized, let's depict what we see. And now you start seeing the sulcus and so forth in here. It's closer to the configuration of a real brain. And here's Gall at a younger age. It's interesting is, we look in the past, a lot of times we always use photos of when they're old. It's always nice to see what they look like when they're younger. All right, and phonology was very popular for many decades. And people looking at skulls to see if they can discern characters of individuals. Here's for presidents, looking at even the chins, which one's more firm. Here should you marry an individual based on your compatibility? We have this, we call it sometimes neurocompanions, which we look at two individuals to see how similar they are. But it's mostly to see where they overlap with problems. Which area do they share an area where there's going to be challenges? Where's complementary functioning? Where one is showing more primitive instinctual behavior, the other one's more reasonable, rational behavior. We can see in the brain 55 areas which ones, how they are, and you can, usually it's a roadmap to where you need to work first on solving the problems. But so the idea has been around for over a century, the idea of trying to map people by their brains or here their skull shape / brains. All right, so well, the one problem with Gall, and in fact, he's the beginning of localization. The idea that certain functions are localized to certain areas in the brain, that the brain is a plurality of organs. Well, that struck some people as blasphemy, because how does the soul come in? If you have a multiplicity of organs, and you got this unitary soul, how does it relate? Do we have to do some kind of calculus to make it distributed well or something? Well, so Florence is one of the first neuroscientists. People would have, there's a few other people before him, but he set up a lab mostly to attack Gall's mind-brain equivalence. He wanted to show that this plurality idea within our head was false. And ironically, he's going to prove the exact opposite. All right, so here's his statement: the unity of the soul, human immortality, free will, and the very existence of God are undermined by this localization hypothesis. So he argued for echopotentiality of cortex. So if you lose some cortex, some area, other areas of the cortex can fill in. Some of that's a little bit true, but not as true as he would like it to believe. So he actually found out by working with pigeons mostly and a little bit on rabbits that if you remove the cerebrum, you lost perceptions, motor function, judgment were abolished. If you damage the cerebellum or removed it, the equilibrium and motor coordination. And if you actually removed or damaged the brainstem, the creature would die. So he showed on a gross level that there was this separation of functions, this localization of functions. One of his issues, and why to this day we talk about when you only use 10% of your brain, it goes back to Florence. What Florence did is he would cut away at the brain of like a pigeon, and he noticed really didn't get certain the deficits in the behaviors he was investigating until he had removed 90% of the cortex of this poor pigeon. And then he would say, in essence, okay, you only need, and that's when you start seeing deficits, so thus we only need 10% of our brain. Well, the problem was he was cutting away the cortex when the functions he was testing were subcortical. They were wing flapping and eating. All right, so had he been investigating, like running a maze or doing some kind of problem solving, like they do with crows, moving pebbles around and doing conservation of mass studies with crows, he would have had immediate recognition that the cortex, you couldn't damage 90% and still get success. But you're doing some very subcortical functions, wing flapping, eating. Sure enough, you don't really need the higher end to do those, to achieve those. Okay, so you can think of the brain as like it's plurality of organs, a mosaic, and in Gall's day, everything was viewed as very symmetrical. We're going to go off of that very soon, where we realize that the left hemisphere and the right hemisphere have complementary functions, but different. And the beginning of that real laterality is, and also validating localization, is a Dr. Bolliard, who had a bunch of patients, over 100, who had aphasia, speech loss, and he noticed that the frontal lobe was always involved in speech loss, the ones of his patients he had. And he was a supporter of Gall, defended him. At his time, the Center of Science was in France at some level, though the Center of Math and Physics would be probably in England still because of Newton. So he saw that language, speech, was the frontal lobe was needed for intact speech, but he didn't notice the left-right distinction yet. There was a country doctor who noticed the distinction, but because he's a country doctor, he had no impact on the politics of his day, on the science politics. I used to have some advisors and people I worked with who would fly into a conference, give a speech, fly out a few hours later, and wonder why doesn't anyone listen to what I say, or follow up my ideas. Science, like every other discipline in the world, is political. You got to have face-to-face contact. You have to interact with them. You got to grab people by the lapels and say, "Follow me," at some level. If you got to do at least metaphorically, and that's why you go into a conference and sometimes get there early and stay there late. Most of the important communications happen at the bar. They happen after the talks that we've been given. So I had it, and it's funny. I had one of my major advisors. He would fly in for a talk, on like a Friday morning. He'd come in and have his talk in the afternoon, and then leave that night. And he just couldn't wonder why no one was following his ideas. Well, you got to be part of the political game, and Broca was a major player in the political game at that time, the science politics. He was the secretary, was the leader of this organization. I think he was a founder of the Neurological Society. So Dax, though, this country doctor before him, finds its left hemisphere damages are most associated with verbal memory deficits. So it's a left-sided issue with the language. His son publishes the memoirs in the actual yearly publication that Broca is involved with, but Broca doesn't get all the credit, and no one's going to know the name Dax unless you're a historian of neuroscience, because Broca, once he proved it, it stayed proved, and that's the same as like Christopher Columbus. It's Leif Erikson discovered America, but it wasn't until Christopher Columbus that America stayed discovered. So you discover something, then no one else follows you. And that idea, it's like it didn't happen. So Broca said, "Nous parlons avec la hémisphère gauche." We speak from the left hemisphere. And he had a famous patient whose name was given the nickname Ton, because all he could say was Ton, who could only say the word Ton, came in, was shown to the group, and then died soon after. With the autopsy, they found this damage right there, and what was soon to be called Broca's area. Ironically, or not ironically, because as you begin to understand how the brain is organized to reflect everything, that area is sort of our political area. It's our hierarchy processing area. The hierarchy needed to do motor programs to do speech, also cognitive hierarchy is done there, and so in a way, it's how we handle the social dynamics of our world is done in Broca's area, and Broca was a person who was very good at social dynamics. Soon after, there was this first area found for aphasia, for speech, for halting speech. There was other researchers on the continent very soon after, you can see in the next decade, who found another aphasia, not those who have trouble articulating, but those who have trouble, with Broca's, it's fluency problems. This would be, they don't have fluency problems, but they have content problems. They just gibber all the time when you have damage in the temporal lobe, which would eventually be called Wernicke's area. And it was discerned eventually that this is the area where meaning is attached or created, or propositions are generated, and this is where they're articulated. So the concepts and conversion to speech begins here and finalized here. So what Wernicke recognized is this model that we pretty much use to this day, a network model, that there's this area here where you start the proposition, and here is where you do your expression of it, and it's a network, and if you actually damage the middle, you'll get another form of aphasia, which he predicted and came out to be true. Predicted, predicated? No, it should be predicted. Conduction aphasia. Okay. So we got this localization, connectionistic model, little hubs coordinating to create greater function. But there's kind of another element going on, and this is where holism can come back up, and that the idea that there's sort of this interactive set of hierarchies. And we do know when someone would have a stroke and lose speech, that there would be recovery. Sometimes it would be some very little recovery, but sometimes it would be major. So we know that other tissue could pick up the job. So it isn't that these hubs are covering a function, handling a function 100%, their neighbors handle a few percent. And if that hub is gone, the neighbors can grow and take up the function, and much of it can be restored, depending on what we're talking about, what function it is and what area it is. So yeah, one of my favorite heroes from the past, John Hewlings Jackson, who recognized that the left hemisphere, one time it was viewed the left hemisphere was everything. In fact, I remember Gazanga thinking that the left hemisphere in humans is the only thing really matured, and the right hemisphere is just simply an ape's ability multiplied times 3 because of the mass is times 3. Believing the right hemisphere is not really developed, but we are successful as a species because of our right hemisphere, not at all because of our left. There's evidence that the common chimp is even more left brain than us. Problem is, you need a balance, and success requires social interaction, peer relationships, being able to understand perspectives of others. That's all right brain. If it wasn't for our right brain maturing, we would still be primitive. So the left actually has kept the chimps in the trees, and the right has created cities. So the leading hemisphere hypothesis by Jackson is recognizing that the right hemisphere has superiority on many functions, such as visual recognition. So again, the right hemisphere is going to deal with the configuration, the uniqueness of a stimulus. The left's going to look for the core category, the familiar of that stimulus. And one of the things people started realizing, when you do have damage and someone stops speaking, doesn't necessarily mean that that's where speech occurs. To localize the damage which destroys speech and to localize speech itself are two different things. Or as Dr. Head says, there was a Dr. Head and a Dr. Brain who studied the brain in the early 20th century, the processes which underlie an act of speech run through the nervous system like a prairie fire from bush to bush, remove all the inflammable material at any one point, and the fire stops. So when a break occurs in the functional chain, orderly speech becomes impossible because the basic physiological processes which subserve it have been disturbed. The site of the breach of continuity, the site of such a breach of continuity, I gotta move this thing away, becomes impossible. I just lost it. The site of such a breach of continuity is not a center for speech, but solely a place where it can be interrupted or changed. All right. So there was that further sophistication in understanding that a damage in one location could interrupt the chain, and thus it's not the function wasn't there, but that was a pathway serving the function. Okay, why is this not moving now? There we go. Okay, so cognitive functions can be localized to a certain part of the brain, and that different functional regions take part in any given behavior. Right? Topographical organization to the cerebral cortex was based on his work with epilepsy. Much of, I'd say the majority of our understanding of the brain initially came from speech problems, aphasia, and epilepsy. Those are the two engines that have driven our empirical development of cognitive neuroscience and all the neuropsychiatric issues that we deal with today. One of the things Hewlings Jackson's have, the reason why I said he's sort of my hero, or was my hero for that time period, is he had a theory called microgeny that kind of vanished for a while. A guy from Princeton wrote a book in the 80s. I read it and then wrote an article and got credit for the theory for a bit, which freaked me out a little bit that just reading a book will give you credit. Well, we probably all have heard the term ontogeny recapitulates phylogeny, meaning that, for instance, when we're in gestation, when we're developing, our brain looks, very much the same. Looks like a fish brain, looks like a reptile brain early in development, and then eventually differentiates towards a mammal and human. Well, the microgeny idea is a very beautiful idea that ontogeny recapitulates our phylogeny, our species history, every moment of every second of every day. That our brain is every brain event is unfolding across our primitive brain going up to our more mammalian to our human level. So that's the way I say it. I'll just read it here. Each brain event incorporates our entire evolution in milliseconds. We scale from fish to human thought every moment in time, except when injury or functional obstacles block us. And often injury will expose earlier stage of processing. In fact, in neurotherapy, when we do a brain map, we can find out where are you stopping your thought process and in which areas are causing you to have more primitive responses. Well, one of the events that led to the recognition of localization of function was Nifas Gage. If you haven't encountered Nifas Gage, I got some beautiful videos. They made a great mini movie, high quality one at learner.org about his case. He was a railroad foreman controlling a team of rough men. One day, while they're dealing with dynamite, explosives, and you have to tamp down the gunpowder with this big rod here. Unfortunately, he got distracted and something happened. There was tamping down, there was sparks, and the thing exploded and shot the rod right through his head, right through pretty much through the orbit of his left eye. Well, he didn't die. And some neurosurgeons noticed, hey, wait, you can cut open this, you can cut the skull, cut the brain and parts of it, and then there won't be death of the patient. That's interesting. But what most people picked up from this incident is as he recovered, he didn't recover fully his emotional regulation. He started connecting to small animals, squirrels, and so forth. He tried to survive the best anyone would after such an accident, but he could no longer control his emotions. And why? Because he damaged his orbital frontal cortex, which deals with governing and inhibiting our behavior in social contexts in particular. So this is the evidence of that emotional regulation is controlled by that orbital part of our frontal lobe. All right, so we're getting these gross investigations or events that deal with brain function. Let's go down to the microcosm. Let's go down, get our microscopes out and see what is the brain made out of. And when you're studying the brain, it's not unlike studying star systems. The only advantage we have over studying star systems is that we can move our optics around. We can move our location. We can slice things differently, where unfortunately we're pretty much locked. All our investigations in the star systems are from Earth. We get the movement of the Earth through the year to give us some investigations into parallax and so forth. But I do think there's an advantage of studying the very small. But it is still very difficult. Studying with the light microscope and even doing the electron microscope, dealing with the very small, it's hard to understand unless you start accumulating a lot of data to figure out what you're looking at. Well, you have Purkinje, who was studying the cerebellum, found this big cell, which is now named the Purkinje cell, with up to 50,000 connections to other cells. Very hypersocial cell. Those are the cells that we use for coordination and governing our behavior in not in a global way, governing moment to moment. Did we do what we intended to do? We reach for a can. Did we actually get to there? Or did our hand grasp it? So forth. That's the governance that the cerebellum does. Then you have Cajol. I don't think it's Roman. I thought it was I got the name wrong. I think it's an A here. So Cajol he's the one who said that the adult brain was fixed, and that once you're an adult, there's no neuroplasticity. And then later said, well, you know what, the future of neuroscience will say if there's neuroplasticity or not. Maybe I'm wrong, and he was wrong. But he was a very major player, early 20th century, investigating the dendrites and the neurons. The idea that there was an issue going on. Should you study these cells or study the capillaries? Well, what's most important for understanding function? The supply lines or the actual units that are doing the firing, the action? And he decided, let's forget about the capillaries for now. Let's just work on the cells in the spines, dendritic spines, and that was probably the best route to go. Yeah, there's his name, Ramon, correctly spelled there. Okay, here's some drawing, and we start recognizing that the neocortex in particular has up to 6 layers, or you can see these 6 little, well, you have the surface layer and then the 5 other areas here of the large cells, and started recognizing that there's this organization of the cells and how they are transmitting information. You send all information. This is the axon firing. This is the receptive field. This is the sending out of the connect axons to the other dendrites of other cells. That would be the receptive field of those other cells. Okay. Well, Golgi was around at that time too. It came up with some beautiful stains that let you see the cells, and some beautiful drawings. And cell types of themselves are very, very interesting. I don't know when the heyday will come where we really understand cell types and how they differ. There's a diversity of cell types that's sort of not understood yet, I believe. Though if people who are studying cell types probably say, no, we all know everything about them. It's like looking at galaxies, different types of galaxies, some level. But I moved to the higher level, which is when we started using what's called Brodmann areas. So Brodmann, using a microscope, started looking at I'm going to show you a slide in a moment, looking at the different areas of the neocortex. Must have done endless amount of work to be even reasonably accurate. He did have predecessors. He'd have people who came after him, but he organized in a way that we often to this day, and we do here at Kaiser Neuromap, we still use Brodmann areas, and I've taught people, hundreds of scientists and clinicians use Brodmann areas. One of the reasons why Brodmann is more successful, his nomenclature. He just used numbers. Brodmann area 1, Brodmann area 2, 3, versus using other types of labels that could be harder to remember. So a lot of success comes with mnemonics, how easy people can remember what you mean. I'm going to show you a lot more in Brodmann though, coming up. Okay, so we know there's this cephalization period. I went over that in the previous masterclass that goes on during gestation. Here's the increasing of the brain heading towards adulthood. Now, our skull will get fixed in size by around age 9 or 10. So the skull's not going to grow anymore. So then now you got locked into this volume, and that's when you start getting more and more convolutions, trying to get more and more surface area in a limited space. This is the proliferation of cells, and you usually get a proliferation and you often get a pruning. It's not shown here, but it depends on what brain area you're looking at and what functions it serve. It will be more cells than you need, and then cells that determined just by basic genetics will create all this cell proliferations, exuberance, and then they'll be cut down as the individual does not need the
neurons that are not necessary for one culture are pruned away, die off.
Okay, so now we're here with Brodmann, and you can see what he discovered is what you call the cytoarchitectural analysis of the brain, the cell architecture. And what he did is we look into the slices of the cortex. And here, for instance, you would see that there's, of those 6 layers of the neocortex, you can see layer 4 was wider in this area for a long time, and then it vanished. And there was a transition from 3 to 1, and he realized, well, you know what? This tissue must serve one function, and this must serve another function. Probably related, probably adjacent in some level, because it's structurally adjacent. In fact, I have a better picture of this. There it is. How to understand function from different structures. For instance, if you were an alien and you had a human in a formaldehyde jar, and you could see the bones of the hand versus the bones of the feet, you would know immediately that this is not weight bearing. This is weight bearing. This is used for grasping or something like that. It has articulation. This one has very little articulation. So from structure, you can figure out somewhat function. And so when he sees structural differences in the brain based on cell distributions, certain cell types like bed cells and the motor strip, and what would be called granular or agranular cortices, he would say that's a different area. So each location be wherever the structure changes, he gave it a new name, a new number. It made it much easier, by the way. There will be people I'm going to show you in a moment who give it a new name. That's why we don't use those people designations anymore, nomenclature. Brahman would just use numbers. So use numbers when you need to. Here he was a great collaborator. He was a synthesizer. He opened up his first book there. He had one 1909. So this has been over 120 years. Because 1901 is he also has a beginning of his work, and he talks about these 6 great neuroanatomists from just the recent generation, the previous generation, and he was trying to kind of incorporate all their ideas. Excuse me, and we still to this day talk about Maynard cells and Bet cells, and those are the names of the people who studied them. He tried to merge them all together, and that may be why that spirit of true scientific collaboration is why we still use Brahman to this day. Okay, so here are the names of the areas, and they have a kind of different. They use a dual labeling system. Sometimes the system area is called by a cell type. So we have a gigantopyramidal neurons in our area 4 that handles motor, and so that is called the gigantopyramidal area. Some it deals with its location. So caudal and rostral, post-central tells you where these are. Pre-parietal means in front of the parietal. The parietal's here. It's actually usually it's still part of the parietal, typically in how we would organize it, but this is our parietal lobe area, and that's the pre-parietal, the frontal point. Here's the cell type and the location, frontal, agranular. Superior means on top of, so superior parietal, it's the top part of the parietal, even though this is higher, but that's the superior parietal. Intermediate frontal, that tells you the location again. The cell type or cell organization distribution, granular frontal, area 9. Frontal polar, that's the location, polar. Prefrontal, area 11. This one's also called the prefrontal. Now, some of these areas like 11 and 12, sometimes you won't even see the level, area 12. Some people have decided it's not enough distinction. They'll merge them together. Other times people will see more areas, and suddenly there'll be an area 13 or 14 in the front of the head. So that's the one thing about science. It's not very clean. We head towards a consensus, but it's always kind of messy. And so sometimes you'll see like area 13 in the front, 14 in the front. Usually, a lot of times, area 13 has now been given to the insula, which you can't really see here. It's in the middle. Striate, that's the type of cells and streaks. And stripes. Peristriate, near it. Peristriate, around it. Inferior temporal, below, down here. Middle temporal or medial temporal. Superior temporal up here. Ventral posterior, so ventral is down, dorsal's up, like a dorsal fin of a shark. Ventral posterior cingulate, so it's the bottom part. You have to see it in the medial view right there. Ventral interior, and these are called the posterior cingulate and interior cingulate. Usually we don't say ventral in front of that, but we usually do say the other if it's dorso-posterior cingulate and dorso-anterior cingulate. Area 32 and 32 is probably one of the most interesting tissue we got. Occipital-temporal, that's the area 37 out here on the side here. Temporal polar, that's the temporal pole. That's where our self, and governs our amygdala along with the orbitals. Our amygdalas are the things that short circuit us. It's a primitive system to detect threat in the environment, but problem in modern day society, it detects it so much, and you get anxiety and so forth from issues, especially when this is not governing well. Angular gyrus right here. Supramarginal gyrus up here. This area 40. 41, the anterior and posterior transverse temporal, or what's usually called the auditory cortices, the primary and the secondary. Apicular, which is part of Broca's areas. Triangular, because it looks kind of like a triangle. That's also Broca. These two are Broca's areas on the left side. On the right side, we usually use those terms. But you usually never even hear these terms because we just say area 44, area 45. Middle frontal is up here. Orbital is here, 47. And then there's all these areas I don't even never deal with because it's so deep inside. We can never really know how they contribute to the EEG. And but all these ones, just this whole glossary of terms. Like this is area 33. I don't even have the number here because it's the pre-genu. This is the genu of the corpus callosum. That's a pre in front of it. There's all these ones in here. And some of these, again, these were found in monkeys only, but sometimes they've been rededicated to humans. So you'll often will see area 13 as the insula, though sometimes you'll see 13 and 14 in the orbital. 27, I think some people even give it to the hippocampus. Sometimes it's the parahippocampus. I've seen different designations depending on which lab you're looking at. Okay, so it's about recognizing that when there's different structures, there's going to be likely different functions. Here's another way of just looking at it. We just looked at before. That's lateral view. This is the view you see very commonly out there. I made a version that's very prominent. This is my version, actually. As you see, that I made focus here on this side and gave these colors what functions they generally serve. I'm not sure. Some is very helpful. Others may not be as helpful. Okay, but he had precursors. Here's Campbell. A lot of this came about from lesion studies, people having brain injuries and figuring out what the functions were, mixed with microscopes. So you can see very similar organization, but a little more gross, doesn't have the distinctions yet. They recognize there's the auditory, there's the visual primary cortex. Smith comes around. Smith just used the wrong nomenclature, almost the same areas as Brahman will use, but horrible nomenclature, horrible names. That's should be a lesson for anyone. You got to name things well. Here comes Brahman in, using numbers, easy to remember. Found approximately 47 areas for the human, depending on which ones you decide, what you label like the insula and so forth. Maybe 48. Then comes along these von Economo, never heard his name pronounced. I don't know how it's pronounced, but finds 102 areas on each hemisphere. Too many areas, just too many notes. So too many areas. People every 20 years or so, there'll be a major publication saying, "Hey, guess what? It's not 50 areas of Rodman that found. It's 100, areas, or it would be 200 for both hemispheres." And I recently had someone send me this article and say, "Oh, that's very exciting," except we've known this since 1925. Too many areas. And again, what you try to do in any science phenomenon is find a point where you're going to get reliable description and ability to communicate with others about what you find. I use the example of like if I was studying baseball fans, if I use a definition, you got to be watching a baseball game right this moment to make you a fan. That's obviously too narrow a definition. If I say, oh, anyone in the United States is a baseball fan, that's too broad a definition. We have to find a reasonable definition. Like, okay, if you've gone to a baseball game, if you watch more than 2 games a year, then you're a baseball fan, something like that. Even in the stands or not, or on TV, or that'd be fine. But you got to find that middle ground, the Goldilocks area. And the problem with this approach, just too many, hard to communicate. Probably wasn't that reliable because when you go across people are going to be slightly organized differently, and then you're going to not be able to find the same phenomenon associated with the areas. This is a Russian version, very similar to Broca, Brahman. And say often you hear Broca and Brahman being mixed up like that. Then you go back. This is the swing. You have a pendulum swing. You have all this, this middle ground. Then you have too much diversity, too many cells, now too few. And you have in 51 saying, oh, you're looking at the brain. You're too. You're seeing distinctions that aren't there, and really we only have 4 distinctions. We got the conio cortex, which is the primary cortex where the thalamus comes in and connects to the cortex. Then you have the isocortex, looks pretty similar around. It deals with one type of sensory information primarily, and then you have the disc granule and A granule up in the front. Not right, clearly not right, but. And here we are with my functional atlas I made. I used to have online. You can look up all the functions that we had papers for. It's still available through the archive, but I'm not updating it. There's all kinds of, much is going on. And whenever I see a paper that deals with Brahman areas, instead of giving me the Greek, like superior parietal, just give me the Brahmin area. Give me both. That's the safest way. And I love people who give the Brahmin areas because you also give what's called this coordinate system one. So I'm going to show you. But here's the insula. The insula was defended against the EEG because if it fired similarly, it would cancel each other out right there. And knew the EEG was coming. Okay, so we get this coordinate system started by this researcher called Telerarch, where it's now an XYZ coordinate space based on where certain items are in the brain. I used the system very early, and they did use Star of David's. My name's David, so it caught my attention. And what it is this is various studies and where they activated and telling you where the various Brahmin areas are. And the three-dimensional system is what most papers will say now. They'll give you a general what we're in, like superior temporal, but this is the coordinates, XYZ. Sometimes the coordinates are Telerarch. Often there was something called MNI, very similar, off by just a bit, MNI right here. So let's see anything that's useful. So it's a three-dimensional coordinate system. Relates all points within the brain to the midline of the anterior commissure and arbitrary point to start our measurement, 0, 0, 0 point. So that's the 0, 0, 0 point. And then it goes millimeters forward, millimeters to the left. That's the positive, and to the right. I mean, this is negative, I should say. So up is positive for Z coordinates. Left is negative. Right is positive for the X coordinates. Forward, interior is positive for the Y coordinate, and negative is posterior. Okay, this is looking at area 45, left and right hemisphere, and so as you can see, it's projected three-dimensional space shown in 2 dimensions using Star of David's. Beautiful study. This is like the 600 papers. If you look online, just type like DK Brahmin areas, and you'll find it. There's 600 papers. Beautiful work. It must have been a dissertation or something like that. Very great help in the history of science. This guy in Denmark, I think. All right, those who love sausage or law should never see how either are made. Well, same is true with science. Like on that page, they do studies, and they didn't want to change any of the data. And sometimes there was a sign mess up, and so instead of a plus Z score, a Z coordinate, I should say, there was a minus. And so instead of putting the activity in the parietal area 7, they put it in the person's shoulder. And they didn't correct it. And I corrected it with skill, and I corrected it with net, which is our current software. So it's a beautiful, here it is. This is a single instance, a single page. This is from like 2006. I don't think it was, anything has been added to this. I would hope people will eventually take over this database, but this shows all the areas that activated, be it in a PET or a bold fMRI study, and what the study did. And it's a very great cross-referencing of like 600 papers. Forgiveness, what areas ahead are involved in forgiveness. Very incredible stuff. This is interesting. This is a summary of how many papers they found mirror neurons in, because mirror neurons are very important. Those are the areas of the cell. Those cells will fire when you do the action or when you perceive someone doing the action. And you can see which areas are lacking mirror neurons, most likely because they weren't found in any studies. Our sense of self, our self-nurturing, our governing our appetites. Which when we have a lot of mirror neurons, speech, perceiving the value of objects, social, context, motion, movement, imitation. All right, so we have all this localization, but of course you have to have someone who says no, because he's the next generation. This way, really about 3 generations back or 4 generations, Lashley, who trained a lot of people, even when I knew, Carl Pribham down here, as mentioned. In his argument, he was working with rats. He was cutting away the cortex of rats, and he didn't see this localization issue. He just saw sort of the more you cut away, the less the functioning in general. But partly it was what he was studying. If he's been studying monkeys, he would have found localization. So, but you always have this, and you still have a little bit of holism out there where people talk about the hologram brains of theories of the brain and the universe itself. They're all a bunch of just hologram with a multidimensional representation, and we just perceive all these dimensions that really are laying on top of each other. Just to mention, Lashley was very important in training people who eventually would lead to the opposite, and it was still in localization to this day and will be for a long time. Other things that came about, Penfield was a neurosurgeon. He looks very suspicious looking at us right now. Like he's not sure if we're going to respect what he's done. He's done a great work. And one of the things is finding what's called this homunculus in the motor and the somatosensory strip. And it showed, this is not a very good picture because it's blurry, but the homunculus will come up later when we go into other functions and the general functions in the brain and the cortex. But it just shows how our body is represented, and the motor versus this. I think this is the motor side, and this is the sensory side. It explains foot fetishism because those areas that represent the foot are right adjacent to the genitals. So stimulate this, you may get cross stimulation in the genitals. That's probably why foot fetish occur. But this is the sensory, shows the face, and the hand is most represented, the thumb. And in the motor, you can see the face and the motor. Compared to the back, hardly has much representation. So it shows the prioritization of representation in the cortex. But there are individual differences when using individuals who have epilepsy, you're figuring out where speech is, different areas have, it's not all uniform. So there is some differences, which makes it confusing. And the final thing I'm going to end on is how we've used rats greatly to figure out what functions mean. And one of the great understanding of studying rats is and how the difference of enriching the situation leads to a better brain. Creating a social area where you change the toys every 2 weeks. You can't let the toys always stay the same. It starts getting boring to the rat. If you change it, you get this enriched condition where you get much better, more dendrites, more complexity in your brain. And you can see all these things that increase. This is enrichment. This is the standard condition. This is the impoverished condition. So this is when you're all by alone, solitary confinement. Your brain shrinks with solitary confinement. This is, you have no toys, but you have other people to hang out with. And here you have the toys and they're being changed every 2 weeks. And I always like to say that, tell clinicians, change things in your waiting room often because to keep people interested in stimulating. All right, that is the history of neuroscience for the Kaiser Neuromap Academy. Have a good week. Bye-bye.